Hello, welcome student. We shall discuss today an application of DNA. Means how DNA replicate. Means in the other words we can say that formation of DNA from RDNA. Understand? This is called replication. This is called replication, means formation of DNA from DNA. DNA से DNA का बनना. That is called replications. One is also that formation of ribonucleic acid, that is RNA, from DNA. यदि डीएनए से डीएनए बनता है तो दैट इज नोन एज रेप्लिकेशन. और यदि आरएनए यदि डीएनए से क्या बनता है, आरएनए बनता है, रिबोन्यूक्लिक एसिड बनता है, तो दैट इज नोन एज ट्रांसक्रिप्शन. तो सो वी सी दैट डीएनए इज द मेन थिंग्स फ्रॉम विच द both nucleic acids, that is either DNA or RNA, is formed. But today we shall see that replication of DNA means formation of DNA to DNA. replication के लिए तीन मेथड्स होते हैं. तीन मेथड replication के लिए तीन विधियां बताई गई है. थ्री मेथड्स फॉर द रेप्लिकेशन. फर्स्ट वन इज कंजर्वेटिव मेथड. सेकेंड वन इज dispersive method, and third one is semi conservative methods. So the three methods we have studied for the replication of DNA. Understand? Conservative methods, in that says that in the conservative method that it deals that new DNA formed directly from old DNA. ओल्ड डीएनए से सीधे नए डीएनए का ये हो जाता है. मान लो कि ये नया डीएनए है, ओल्ड डीएनए है, और इसे सीधे जो है ओल्ड डीएनए, ये न्यू डीएनए और ये ओल्ड, यानी ओल्ड से सीधे नए बन जाएंगे. समझे? तो इसके लिए कोई नहीं बताया गया, तो दैट इज नॉट एक्सेप्टेबल. डिस्पर्सिव मेथड, that DNA strands splits haphazardly and combine to form new DNA. डीएनए का जो स्ट्रैंड्स होता है वह टूट जाता है, फिर कॉइलिंग अनकॉइलिंग होते हुए फिर वह नया डीएनए बन जाता है, समझे। तो दैट इज known as dispersive method. We also study, but it is not generally acceptable. But these two methods we use, we accept, and which is much acceptable method is semi-conservative method, which we shall discuss now. Now, what in replication takes place? We know that DNA is a double-stranded structure. This is a double helix structure, that is DNA. Understand? First, there is initiation of replications, and next, after that, it will bifurcate. Means both strands will be separate from each other. They will be called template. They will called template. Understand? And now the complement. This is the complement strands forms. Understand? This is also so one is, this is the actually template, and this is the and this one is complement. Understand? So in this way, a short description that the replication from one DNA to, means one is template is actually parental, and this one is newly synthesized. Now in that is, we can say that इसको हम ऐसे भी देख सकते हैं, these are the bonds base A, T, G, C. This is T, A, C, G. Understand? Now these are the bases. Now they bifurcate, that is they differentiate in the same way, that is A, T, G, C. This is called T, A, C, G. Understand? There are the template of parental case. Now the newly synthesized will its complement is formed in that kernel 3 prime 5 prime, that complements are formed, and means complements of T is A is T, C is G, G is C, A is T, T is C, G is C. Now these fragments are called Okazaki fragments. Okazaki fragments. So here is bifurcation, that is enzymes we shall discuss in the next video, helicase. Next one is here isomerase. Topoisomerase, the alternatives are forms, and finally they join each other, that is by means of ligase. तो जो सेल है topoisomerase plus proteins, these all are combined and forms a newly synthesized strands, and that type of strands means now they will separate and forms a new DNA in which इस तरह आप दोनों अलग हो जाएंगे और अलग हो जाने के बाद इस तरह जो डीएनए बनेगा जिसमें एक पेरेंट्स का स्ट्रैंड्स होगा, एक न्यूली सिंथेसाइज होगा, एंड यहाँ पे एंड दैट टेक्स प्लेस यूजुअली in the S phase of cell division. In the S phase, you know that the amount of DNA becomes double. That replication takes place. Sometimes it is asked where the replication takes place. That is especially at the when the amount of DNA becomes double in the S phase during cell division. That is in the interphase of S stage. So it is a just outline of the replication of DNA, and if the in place of T, if the U is applicable, U is formed, that will transcription which will form in the formation of ribonucleic acid in the next video.